Here is another look at the control elements. If you look at this blue strand at the top, again, this is going to be the DNA template. The yellow pieces on the left are still the control elements. Now you see the name distal, and what that means is it's much upstream to the promoter region. If it said proximal control element, that means that non-coding DNA genes would be closer to the green, which is the promoter region, but they're the same thing. So control elements are going to be the non-coding pieces of DNA where the transcription factors, those proteins, can bind. Now in this picture, the proteins are called activators. So these are going to help increase the likelihood that transcription can occur. So this would be known as an enhancer region. So lots of proteins are involved with this transcription beginning. So these activators are going to bind to the control element or the enhancer region, and it allows for the proteins to bind to other proteins. So protein-protein interactions are really important for transcription. So in the second picture, you see a blob of different purple pieces. Those are proteins, and they're known as mediator proteins. So those are going to help get RNA polymerase to that promoter region for transcription to begin. And they bind to the activators, which also bind to other proteins, which will allow, which I think I just said, RNA polymerase to begin its job of transcription. This diagram compares DNA strands that are in different types of cells. So on the left, we have a liver cell or a hepatocyte cell, which in our liver cell, liver is responsible for detoxifying. And one type of protein that liver makes is a type of blood protein known as albumin. And on the right, we have a lens cell. So this would be a cell located in our eye, and it makes crystalline, which is a type of eye protein. So why is it that, again, we have the same DNA in the liver cell and the lens cell, yet the lens cell is going to produce crystalline and the liver cell is going to produce albumin? So the reason, again, has to do with the enhancer region and specifically the control elements and those transcription factors or activators that are going to bind. So let's compare. Looking up at the top, you can see there are different control elements, so non-coding pieces of gene where those transcription factors or activators are going to bind to. They are upstream from that promoter region, upstream from that TATA box, and where transcription is actually going to begin in order to ultimately code for those proteins that I just mentioned. So down at the bottom, on the bottom left, you can see this would be a liver cell, and the only activators or transcription factors that are present here are going to be those in which allow for albumin to be synthesized. In a lens cell, we don't have those activators present. So please review the slide before to understand this process if it's still a little bit foggy. And on the right-hand side, you can see that of the available activators, only those are present that are going to help to make crystalline. You will not have any activators associated with albumin. So that's why the lens cell does not produce albumin and the liver cell does not produce crystalline. It specifically goes back to the activators available that are going to bind to the control elements in that enhancer region upstream of the promoter. Alternative RNA splicing is the reason that we can have the same strand of DNA do transcription, and the way the exons are put together ultimately can code for different polypeptide chains, different types of proteins, many different types, based on how the exons are put together. So this picture at the top, again, in blue, you see the DNA, you see the exons that are going to stay in the mRNA strand. The piece right below is pre-mRNA. So there's 5 different exons. The introns are going to be spliced out. And just a simple example, on the bottom left, you can see the exons that stayed are 1, 2, 3, and 5. And then on the right, 4 different exons stayed, 1, 2, 4, and 5. Again, mRNA is going to be translated, and then the polypeptide is going to be built. So if the mRNA is spliced together differently, then it will ultimately be a different protein, all originating from a same strand of DNA.